Hello, everybody, and welcome back to the I-305 lectures. I'll do this video recording of the SNP lecture, one of the two lectures that is remaining. And before I dive into the lecture itself, I wanted to clarify a few aspects about the course delivery. So, first of all, the NGS quiz, there was some confusion last night. People were emailing asking when the quiz is going to happen. So, like I posted yesterday, the quiz is was not supposed to happen in the lab, even if we didn't go on this, if we didn't have this whole virus situation, we would still be performing this at home. It's a self-sufficient self-study module that people have to go over individually at home. So the quiz will be posted for everybody on Thursday, tomorrow that is, and you will have a whole day to complete it. I know some of you, again, a few people already messaged me. Some of you are having some sort of an assessment around 1 p.m., so I will open it in the morning and I will leave it available for the rest of the day. So you have the whole day to complete it. About 15 questions, multiple choice or multiple choice. Speaking on the multiple choice questions and the quiz, some questions from about 5, 7 people I received yesterday about the second midterm. People were supposedly not happy with the amount of time. So just for your information, you are allowed to have 1 minute and a half per question, per multiple choice question. These are the standards. So we had 25 questions, I believe, and I gave you 33 minutes. So if you take the 33 minutes, divide by 25 questions, the average amount of time per question is 1 minute and 32. So if you multiply it by seconds, that's 80 seconds, and that's pretty much 1 minute and 20 seconds. So slightly less than the standard amount, 1 minute and 30 seconds. The reason I gave you slightly less is because, let's get serious. You are not necessarily under, strictly controlled environment. You're still at home. You still can potentially have multiple computers, multiple, obviously, sources of information. So you're getting less time because of that, slightly less time. We're talking, 10 seconds less per question. And all the questions are exclusively multiple choice. There are no short answers. There are no calculations where you have to, spend extra time solving them. And again, let's get serious. The test, the second midterm was pretty easy. It's only 2 lectures covered. It's not a cumulative test or anything like that. So I wouldn't expect, getting more than 1 minute 30, 1 minute 20 per question. For the final exam, since it's going to be cumulative and obviously it will be worth a lot more marks, I will allocate more time, but I wouldn't expect to have more than approximately 1 minute and 40 seconds, 45 maybe seconds per question. So keep that in mind. As far as the connection issues, some people emailed me saying that they had their internet crashed. So first of all, I see all these emails, and some people are sending like 20, like literally 20 emails. Don't do that. I have a lot of students. There's a lot of questions, and specifically about internet crashing, I can't answer that question right now because I need to consult with the chair and see what we're going to do about this. Some people are clearly having these issues. Some people, who knows, might be, trying to fool us. But the idea is to come up with a standard approach to this. So I don't know what we're going to do about the people who had their internet connection fail during the test. Once we come up with a solution for everybody, I will let you know. So don't worry about it for now. You're not going to get a zero. You're not going to fail the test or let alone the course just because of that. It's just we need to, given the circumstances, we need to come up with standard solution across the board for everybody to be happy and everybody to be, I guess, on board, for all the instructors and profs to be on board. So just take your time, study for the remaining of your, for the rest of your assignments, for your quizzes. Some people were claiming that the grades were not automatically transferred into their grade book. Again, like this is a super minor issue for this course or for any other course. Do not go crazy. Do not panic. You're not, if you completed the quiz, especially if you completed the quiz, right, or whatever the test, the midterm, and your friends, as some of you were saying, my friend got, the grade transferred immediately, and I'm not seeing my grade. So the quiz has been taken, the midterm has been taken, the grade is there, unless you had, like I said, an internet connection issue. The grade is there, so even if you didn't get transferred right away, it's not a big deal. I will have to transfer them manually if they haven't been transferred by the system by now. Sometimes it takes a little bit of time. There's a delay, there's a huge load in the system these days. You got to understand all of these things. So the minor technical issues like that, like I said, for this course or for any other course, don't have to, panic. You don't have to send 50 emails to your professor. It just nothing is going to get solved super quickly. It will take some time for multiple reasons. But at the end of the day, week or so before the grades are due, you'll see all your grades, especially for major assessments like midterm. Everything is going to get updated. Everything is going to be there. If some grades will be missing, you can always email Van and say, the grades are missing. We got to fix that or something. Before, speaking for myself, before I finalize your final grade, I look at everybody on the list and I make sure nobody has an empty grade or a zero for no reason, unless the person forgot to submit the assignment or something. But if you're missing the grade, but you actually took the quiz or took the midterm, don't worry, everything is going to get updated at the end of the day. And for the quizzes, the grades are in the system. I can see them. Apart from those few people, it's not that many. It's about 3, 4 people or something like that who actually had internet issues. So for those people, we will figure something out, and I'll get back to you as soon as I know myself. Okay. So just wanted to get that out of the way. So for the lectures, like I said, today we will do the SNP lecture. It's not a big one, but it may appear to be fairly complicated or somewhat complicated for some of you. I'll comment on this in a second, but take your time, study well, and there will be a second lecture eventually posted on barcoding. The barcoding lecture is going to be very easy and pretty short. And I strongly suggest you use that lecture. What I will try to do is I will explain the theoretical material in the fish barcoding lecture. And I will also comment a lot on the lab stuff that is supposed to happen next week, week 13. So the fish barcoding lab. So before you take the quiz next week on the fish barcoding lab, you need to do two things. You will need to study the materials that were posted, and you will need to listen to the lecture that I will post eventually as well. Again, that lecture is going to go towards your final exam as well, but it's going to be short, simple. It's not really that much to explain about the barcoding. Nothing like with the SNPs lecture. And this one here, it's going to be a very easy one, pretty straightforward. Most of the information you already have access to. It's that file that I posted in eCentennial, a bunch of files. So I will just explain it one more time. I will walk you over the lab. I will walk you through the lab, and you should be able to take the online quiz for the fish barcoding lab easily. The fish barcoding quiz will be online, obviously, again, and those quizzes are being designed by the fish barcoding quiz will be designed by your instructors individually. So each section will have their own instructor design their own quiz for you. So if you have any questions about the lab quiz, the fish barcoding quiz, you should be emailing. The best person to contact would be your lab instructor and not myself. Instructors are designing, like I said, individual quizzes, and I sometimes take a look at them, sometimes I don't. So I may not know what's going on, but the format, the standard format, is the same for across the board. It's going to be mostly multiple choice. Some, maybe a couple short answers, like really short answers, or like the fill-in questions, but again, really short ones, but mostly multiple choice. One more thing about the files, the lab files posted here. So somebody brought to my attention last night, if you go to the next generation sequencing file here, this module here, if you scroll down, so you look over these, this video I think is dead. I posted some additional videos. So if you look down here there's a paragraph here stating that once you completed blah, 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 all this module, you will have 40 minutes to complete the 20 multiple choice quiz. And then it gives you the date. So completely disregard this. This is just an artifact from the previous semester or whenever, whichever semester that file was taken from. And also disregard this link. This is just an artifact from before. So I will post a quiz. Do refer to your primary source of information about the quizzes should be the news section. Once, especially for the NGS quiz, I will be the one posting it for everybody. So like I said here, I'll give you the time. It will be tomorrow, sometime maybe early in the morning, maybe a little bit later. I will post the quiz and everybody will have to complete it. So that quiz that I just showed you, that link that I just showed you, just completely disregarded. So I hope that clarifies most of your questions, organizational questions, I guess, about remaining lectures and the labs as well as the midterms. Again, I will get back to you. Don't worry about your zeros for the midterm. If it's an internet connection, we will sort it out and we'll come up with some sort of a solution for you. And on to the lecture now, I guess. So the SNP lecture, it probably is, like I mentioned earlier, one of the harder ones, but it doesn't mean that it's super hard or anything like that. It really depends on your level of molecular biology preparation, I would say. So how well you understand the basics of the genetics, how well you understand the basics of some molecular biology. And when I say basics, I really mean just the basics. We're not going to dive deep into really complicated aspects. It's just there's a little bit more details than you would usually expect from the lecture. And given that it's an advanced biotechnology course, it really this lecture here really covers a lot of essential concepts, genetical concepts that you definitely need to be aware of nowadays. So the lecture might require, as you study, it might require some brushing up on your genetics knowledge and maybe some other molecular biology concepts. I spent a lot of time designing this lecture, taking out not essential information, putting in some additional information to help you understand the concept. It really took a lot of time to design. Again, given slightly elevated level of complexity, given the fact that we don't do this in the lecture and we do it online, so you're limited in your ability to ask the questions. So there's a lot of information here. I hope you won't need to refer to other resources much because all the essential information, all the essential concepts I explained here. The topic itself is out there is pretty complicated, so do not try to use really, advanced resources. You might get completely swarmed. Just follow whatever the lecture has, and maybe brush up, like I said, on certain concepts, certain basic concepts, if you feel like you're still not getting them after my explanation. So definitely I would say, consider doing it. Consider looking it up if you need to as you go through the lecture. Otherwise, if you just might have difficulty getting a grasp of what's happening here. But again, don't get scared. It's not that complicated, but maybe a little bit harder than usually. It really depends on your level of preparation again. So do not expect anything super fancy, but nevertheless, pay attention. So what we'll do in this lecture, we will cover the essential basics of the SNPs. We will define what the tag SNPs are. For example, what is the haplotype? What kind of concept is this? And what is it being used for? Some of these concepts, like SNPs themselves, as well as the tag SNPs and the haplotype, I would imagine are fairly new concepts that you're unfamiliar with. We will look at briefly at what kind of effects the SNPs have on the protein structure, as well as function, and what kind of impact the SNPs have on other aspects of the cell function and the cell operation. We will briefly look at the application for the SNPs, the application the SNPs have in medicine as well as in research. A lot of these applications are actually related to medicine. That's where they really shine, but not just that. We'll also touch briefly upon such aspects as pharmacogenomics and pharmacogenetics. But the application, I have to say, the application of SNPs is pretty straightforward as long as you understand the theory behind how what is SNP and how exactly people are using them to do whatever is needed to be done. So your understanding of the application heavily relies on your understanding of the basics of the methodology and the basics of the genetics that lie behind the SNPs. Uh, as you go through the lecture, always ask yourself if you understood the concept that was just described. Always ask yourself why is something happening the way that it's happening. So don't be shy asking the question why. Don't just take things for granted. It might kind of snowball, and you might find yourself struggling understanding a little bit more advanced concepts. We're not going to, like I said, we're not going to go into any super complicated details that this topic is very rich on. We're not going to do that. It's too much. We don't have enough time. We only have so many weeks left, and there's no lectures in the class anymore, so we got to be careful. But again, feel free to use additional resources if you feel like you need help understanding some of these concepts. So before we start discussing the SNPs themselves, some basic, I'd like to introduce some very basic genetics terminology that everybody should be super comfortable with. We'll start from the very bottom of the list here. A locus. Make sure you know what it means. Locus is something that I like to refer to as a geographical position of the genetic element, whether it's a gene or whether it's any other genetic element. Remember, it doesn't have to be the gene all the time. There's lots and lots of the genes are actually an absolute minority in your genome. So it could be a regulatory element, it could be some sort of a repeat, it could be some sort of mutation, and so on and so forth. So geographical in this context is obviously in quotes here, as you can see. So don't take it literally, but in a way, you can definitely think of the locus being like a home address for the gene or for the genetic element. The locus would define the position of the gene on the chromosome. So whenever you describe the locus, whenever you describe the position of the gene on your chromosome, you would have to indicate the number of the chromosome. You would also have to indicate whether the gene is located on the short or the long arm, and you would have to state a specific region and subregion in some cases of the chromosome that the genetic element under study is located on. So in a way, like I said, it's a very good analogy to say that the locus is kind of like a home address for your genetic element or the gene. So that's that. The next one, the polymorphism. The polymorphism is basically any heritable difference in DNA sequence that you can come across among different individuals. So any sort of difference, whether it's a SNP, which we will discuss in this lecture, or whether it's a multiple nucleotide difference. So when you have several nucleotides differ between two individuals, whether it's an insertion, whether it's a deletion, or maybe a tandem repeat, as long as there is a difference in nucleotide sequence, those differences fall under the category of polymorphisms. Polymorphisms are often looked at together with mutations, and it's actually a common source of confusion in scientific and medical literature as to what we call a polymorphism and what will fall under the category of a mutation. These concepts are indeed pretty similar, and I would say there are two major ways to differentiate, two major, I guess, approaches that people use to differentiate these two concepts, and I will briefly talk about both. So one way to look at the differences between the polymorphism and the mutation is to look at it from the medical standpoint of view. Polymorphism from the medical standpoint of view can be looked at as a benign change or benign difference among the two or several DNA sequences coming from different individuals. So as long as that change is benign, as long as that change doesn't lead to any medical conditions or any weird phenotypes that might compromise the fitness of the individual, that is considered to be a polymorphism. On the other hand, if you have a change between the two sequences or maybe more sequences, and that change is actually responsible, directly responsible, or somehow involved in generation of a diseased phenotype, some sort of a medical condition or some sort of a nasty phenotype, then that change can be referred to and usually is referred to in medical literature as a mutation. So mutation has this negative kind of negative, I guess, taste to it. In a scientific literature, in a more not so medical literature, there's a slightly different approach to these conditions and to mutation and polymorphism. And mutation is being looked at as more of the means for the generation of the polymorphisms. So in other words, the mutation occurs in your genome, and that mutation leads to the formation of the polymorphism. As simple as that. Again, so you can have, let's say your DNA gets exposed to UV light or some mutagenic chemical, nasty chemical, or maybe potentially a DNA polymerase slips during the during the replication of your DNA, and that leads to the generation of the mutation. Okay, so once the mutation is generated, that mutation is basically a mechanism for the formation of the polymorphism between the two individuals. So that's the more, I guess, scientific approach to the differences between the two. Lastly, one of the main concepts you absolutely need to understand is the allele. And an allele, you should be pretty familiar with this term. Should be pretty familiar for most of you. Allele is just an alternative form. It's a variant of a specific genetic locus. Again, it could be a gene or it could be just a general genetic element of some sort. Like I said, you need to know that genomic DNA, the majority of your genomic DNA does not actually encode for any proteins. So it's actually absolute minority, in fact. So you have two alleles. You can have potentially two alleles for each gene. One comes from your mom, one comes from your dad, and depending on the identity of these alleles, depending on the identity of these alternative forms of the genetic locus, which could be the gene, you can have different types of genotypes, and we refer to those genotypes as homozygous and heterozygous. So again, pretty basic. If you have two alleles that are the same, you are homozygous for that gene, for that locus, if you want to be more general. And if you have different alleles coming together in your genotype, then you are looking at heterozygous genotype. That genotype is called heterozygous. The homozygous genotypes can be also classified as recessive and dominant. The alleles can be classified as recessive and dominant. So again, I kind of expect you to know the differences between these. So we're not going to spend time going over these. This video here, as it says, it's optional, but it's very good. It's done by a very famous scientist, one of the people at the who was at the beginning of the SNPs research, at the start of the SNPs research. So he tells you how the SNPs are identified, how are they necessarily being made sense of, and how they are being employed for medical research. I definitely suggest you watch it sometime, this video, especially given how everybody has a lot of time these days. But as far as the final exam is concerned, this video is 100% optional, so there will be no questions on this video on the test. So what are the SNPs? First of all, you absolutely need to know what the acronym stands for. So the SNP stands for single nucleotide polymorphism. So it is a polymorphism. If you look at the definition, right, if you look at the acronym, it is a polymorphism. So it is a difference between the genotypes of two or several individuals. And in this case, in case with SNPs, these are specifically single nucleotide polymorphism. In general, polymorphisms can be made up by several nucleotides. It doesn't always have to be a single one, but in this specific case, for this specific class of genetic elements, the nucleotide has to be a single one. So it has to be specifically single nucleotide variant within a gene or genetic sequence in general. So in order to identify the SNPs, what you would do is you would extract the genomic DNA from two individuals like you have here in this example. It can be actually several individuals. It doesn't have to be two all the time. It's just the simplest case when you take just two people, sequence their genome, and kind of compare side by side like that. But in the real world studies, usually people obviously sequence and compare the genomes of multiple individuals. They usually look at them in groups, major groups, and subpopulations, and so on and so forth. So you would isolate their genomic DNA, you would sequence their genomic DNA, and then align it side by side. Align the obtained sequences and to be able to properly compare them for any sort of changes, for any sort of mutations. So in some cases, like if you look at the sequence and that sequence, the first nucleotide is the same, it's G. The second nucleotide is the same, it's T, and so on and so forth. But in some cases, you can have, you will observe the single nucleotide changes within the sequences. So as in this example, C for the first guy, for the first variant, gets substituted for T in the second variant. Pay attention that these happen at the same positions. So the sequences have been aligned, and these are the same positions. You're not just comparing randomly the C to a random position in the genome. They are the same positions on the same chromosome. So this kind of a change would be classified as a single nucleotide polymorphism only in one case. It has to be a single nucleotide, and it also has to occur, as it says here, it's a major criteria, it has to occur in at least 1% of the population. Okay, so one or higher. And those kind of SNPs are called common SNPs, which means that this nucleotide here, this variant here, which is a T, if we consider this to be our wild type and this is our SNP, a variant of the SNP, so this variant here has to occur in at least 1% of the population. If the rate of occurrence is less than 1, is under 1, so in this case it would not fall into the category of SNPs. Or sometimes they call them the rare SNPs, and they're not being studied as much. But the standard criteria is 1% of the population. So the variant, if we consider this to be our standard phenotype, genotype, the variant T in this case has to occur in at least 1% of the population. So know this for the test. I might have a question, on the test straight up asking you, what is the lower limit for the occurrence of the mutation for it to be classified as SNP. So make sure you know it is 1%, at least 1% or higher, right? Couple more numbers associated with the SNPs you need to understand. These three numbers here, 4 numbers at the bottom here. I'm not going to be testing you on these, but make sure you understand well what they stand for. So if you take two random people, and if you were to sequence their genomes, all of the nucleotides I'm talking about in the genome, and that's about 3.5 billion of nucleotides, you would find that two individuals differ from each other only by 0.1% in their genomic DNA. So 99.9% of your DNA code is exactly the same between two individuals. And all this phenotypic difference that you observe around you can be attributed to such a small number, such a small percentage, only 0.1%. So if you were to look closer at these differences, at these 0.1% differences, you'd find that about 90% of these differences between people are attributed to SNPs. So you and your friend differ by 0.1%, and 90 plus percent of those differences are because of the SNPs. So that tells you something right away about you know the importance of the SNPs, potential importance of the SNPs to the medicine. This number here, it says 90 plus. This number here varies anywhere between approximately 85 to 98%, depending on the source. So some people claim SNPs are actually contributing to as much as 98% of the difference between our genomes, but we'll just stick to this kind of average value of about 90%. So quite a value, like I said, quite a bit. Most of your differences are due to SNPs. And like I said, almost all these differences, genomic DNA, can be, or most of them ideally, can be utilized for medical applications. It is estimated, this number here, it is estimated that the SNP occurs approximately every 500 to 1,000 nucleotide. So if you were to recalculate it, we have about 10 to 30 million SNPs in the human genome. Again, the number varies a little bit. Some people claim it's closer to 30. Some people claim it's closer to 10. So I tend to believe it's somewhere in between. So let's just remember, keep this range in mind. You don't have to know it for the test. I'm not going to be asking you specifically any questions on this value, but definitely be aware of, I guess, the order of magnitude of these values. It's one of those biological constants that you kind of it's kind of not a bad idea to memorize those, remember those. How many genes we have, how many SNPs we have, and so on. This number, however, stating it again, you will be tested on, so make sure you know this 1%. So if as much as 90% of the variability is attributed in a genome, can be attributed to SNPs, it means that the SNPs are probably, not the worst candidates, to say the least, for applied medical research. And that's definitely the case. The SNPs have been looked at for the longest time, and they are truly are, great candidates for the medical research. So much information can be fished out about your genome, about your phenotype, about your different sorts of traits and susceptibilities and sensitivities based on your SNP profile, and we'll discuss that here. So a lot of medical research out there for several decades now has its goal to basically link specific SNPs to specific known medical conditions, specific diseases, and maybe in some cases, maybe distinct, unique phenotypes in general. It doesn't have to be the disease all the time. It could be just some interesting phenotype. For example, like the one you looked in the lab. We will talk about it a little bit here as well. Your ability to taste PTC chemical, whether you can sense it, whether you can taste it as a bitter chemical or not. So, not a disease, obviously. Don't get freaked out, but it's clearly an interesting phenotype, and that can be apparently attributed to SNPs as well. So, and that's what we're going to be mostly concentrating in this lecture upon. How the researchers actually doing it? What are the different approaches to linking the SNPs to specific medical conditions? It's not a very easy research. It's not a very straightforward research. However, it's not that complicated. There's a lot of math involved in it, but we will only scratch the surface of that math. So what is the methodology? What are these different direct and indirect approaches? What is the methodology, and so forth. So potentially what you can do is you can find the diseases linked to certain SNPs, and you can identify those SNPs, for example, that are linked to different levels of response to the drugs. If you happen to have say a C in a certain position, you could be more sensitive than other people to a certain drug. If you have a certain SNP in your genome, you can be showing some side effects towards a certain drug, or you could be completely insensitive to that drug, and so on and so forth. So, and once you link those SNPs to those conditions and to those interesting phenotypes in general, what you can do is you can utilize the SNPs as genetical medical markers, as genetic, or sometimes they call them medical markers. Even in forensics field, you can actually employ SNPs to help the police identify the potential criminal by fishing out, some phenotypic information about that criminal, some phenotypic traits about that criminal based exclusively on his SNP profile, the SNP profile of the suspect. So it's a really, there's a lot of really interesting applications for this concept. One very important aspect that you shouldn't be forgetting in the context of the SNP discussion, and I think we discussed this previously in the I-305 course, and definitely I remember we talked about this in pharma. Please remember that not every change in the DNA sequence necessarily manifests itself on the protein level and therefore on the phenotypic level, meaning that you can have a mutation, right? You can have a polymorphism in your genome, but no change in the protein structure will be associated with it, or rather, it will be that mutation will not cause any changes to the protein structure. Make sure you know the molecular biology reasons behind such an event, right? I think we discussed this in BI 305 before we went on this vacation. One of the main reasons behind the mutation not leading to the change in the phenotype is because of the redundancy of the genetic code. Make sure you remember what that is. The concept which implies that the same amino acid can be encoded by multiple different codons. Remember, each amino acid is encoded by 3 nucleotides, and some of those amino acids can be encoded by different codons. So in cases like that, a real and observed, change to your DNA structure, so a SNP, may still lead to exactly the same amino acid getting incorporated into the structure of your newly forming protein chain. Even though you have a mutation, the amino acid remains the same. So clearly, no phenotypic change will be associated with such protein, and no phenotypic change will be noted. Another reason for the SNPs and mutations in general not manifesting themselves on the phenotypic level has to do with the chemical nature of the amino acids that are making up the protein. Remember, some amino acids are acidic, some are basic, some are neutral and hydrophobic. All of that is governed by the nature, or rather the identity, of the side chains of the amino acids. So for that reason, in cases where you have, say, an acidic amino acid getting substituted by yet another acidic amino acid, or the other way around, the basic amino acid substituting another basic amino acid, or hydrophobic getting swapped for another hydrophobic, it is not very likely that you will observe a notable difference between the two proteins on a phenotypic level, because you're substituting like for a like, right? I said it is not very likely. That means you're not completely guaranteed that even if you substitute, acidic amino acid for another acidic amino acid, or basic for basic, you're not completely guaranteed that you're not going to see any changes, but you are unlikely to see that change. You're unlikely to see that change. It all really depends. The results may vary, and ultimately, whether you see that change or you don't depends on the final impact that the new incoming amino acid is having on the structure, on the local structure of the protein domain, or whatever the active site, and therefore on the overall activity and potentially, the conformation of the protein. In other words, you can have acidic amino acid substituting another acidic amino acid, but even though they are similar in their chemical properties, that second amino acid could still be affecting the structure of the active site significantly, which will lead to completely altered activity of that active site and therefore enzymatic activity of the protein as a whole. So, but most of the time, if you're substituting, acidic for acidic and basic for basic, especially if it doesn't happen in the active site, then you are unlikely to see any, phenotypic manifestation of that change. And lastly, remember that the impact of any mutation, and I think I already kind of touched upon that, the impact of any mutation or the polymorphism, and SNPs are not exceptions here, absolutely not. They are just another word for your mutation. So the impact of the mutation, the impact of the SNP depends on the localization of that mutation. Remember, I gave you the analogy in class. I think definitely gave it in pharma, probably gave it in BI-35 as well. If I damage the marker, the marker that we used to write on the whiteboards, at the bottom end of it, the part that comes into contact with whiteboards. So clearly, the quality and the functionality of the marker is likely to be severely affected. On the other hand, if I, chip off a piece of plastic from the opposite end of the marker, it's unlikely to have any impact on its functionality and its quality. So exactly the same concept applies to the proteins. If the mutation or the SNP we're looking at has occurred in the active site of the enzyme, right, the site that is directly involved in bringing the chemicals together and setting up the chemical reaction in place. So then clearly the chances rather of that enzyme having its functionality affected. In some extreme cases, you can have complete loss of functionality, for example, or you can gain a new functionality. So in this case, the chances of the functionality getting affected is very, very high. While if the mutation occurs in some structural but not necessarily essential part of the protein molecule, you are unlikely to notice that mutation, that polymorphism manifests itself on the phenotypic level. This article here, the original SNP article, again, it's optional. You can take a look at it if you're interested in genetics and a little bit more, but it's definitely not going to be tested on the final exam. So it's just your kind of self-study or self-interest, I guess, resources. So moving on to some additional facts about the SNPs that are kind of essential for the future discussion. If you think about it, it's only about 2%, right? Estimated about 2% of the genomic DNA that encodes for the proteins. The rest of it, about 98, is being commonly labeled as so-called junk DNA, right? Obviously, there's a lot more to the story than just that DNA being junk and playing no role. It's definitely not the case the more we learn about the DNA. But at least we can say that that type of DNA, that 98% of the DNA, does not encode for any proteins, right? And that's kind of important for our future discussion. Why? Because since most of the most interesting mutations or SNPs, as you can imagine, are the ones that occur in the genes, right? And therefore lead to phenotypic changes associated with those proteins. They affect the gene gets a mutation, that mutation affects the codon, the codon affects the nature of the amino acid, that amino acid gets swapped for another one, and you get now a substitution that may lead to significant phenotypic changes, right, or functionality of the protein. So if most of the since most of the interesting mutations are the ones that occur in the genes, and the genes are actually constituting about 2% of the genome, the valid question that you could ask is, does this mean that 98% of all the SNPs pose no interest to the researchers, right? Why would we care about all these SNPs that are segregated outside of the genes, outside of the sequences that encode for the proteins? Technically, we shouldn't be really interested in those. It's a pretty logical assumption, I believe, but and that's what researchers definitely asked themselves when they were first looking at it. But luckily, I guess for us, it turns out that this is not quite the case. So the statement is not quite true. And that's actually where the most application of SNPs uh, comes into play. Um, how does this happen and how is this possible? So if we widen our understanding of the gene a little, and if we consider a gene as not just an open reading frame that encodes for the protein, but an open reading frame plus about 10,000 kilobase pairs upstream of it, then it turns out that about 98% of the genes are segregated within just 5,000 base pairs um, of at least one SNP. Okay, so as many as 98% of them are within 5,000 base pairs of at least one SNP. Some more stats down here. 93% of the genes in your body actually contain at least one SNP. Almost 60% of the genes have at least five SNPs or more, and as many as almost 40% of the genes have at least 10 SNPs. So as you can see, it seems like that SNPs prefer to segregate in the fair proximity to the genes, not necessarily within the genes themselves, and not within the open reading frame itself, but around the genes. It is, in fact, not only a very interesting, but also a very useful observation and finding that was eventually put to use by smart people out there, by the researchers. Um, this kind of SNPs that we're talking about, instead of being, the actual causative agents for a specific medical condition or the phenotype, like for example, mutation in your genomic DNA is the famous example from the lab. A mutation in your genomic DNA is responsible for you being unable to taste BTC, right? So that mutation, that SNP in your genome, is directly responsible for a weird phenotype or unusual phenotype where you are unable to taste BTC. Or I could give you a medical example when a mutation leads, for example, to a sickle cell anemia, when the red blood cells lose their ability to significantly get their ability to transport oxygen affected. So, but these kind of SNPs that we're talking about here, the ones that are segregated around the genes, not necessarily inside of the genes, but around the genes, can be still successfully utilized as so-called genetic or medical markers. So, and the key word here is marker. So that gene, or rather that SNP segregated around the genes, is not a causative agent for a condition. It means that SNP is not directly involved in changing the protein structure. So even if you were to hypothetically fix that gene, or if you were to hypothetically fix that SNP, you wouldn't be able to fix the medical condition. But we nevertheless can still use these SNPs segregated around the genes as medical markers. We can use them, I like to use the analogy of the road signs of sorts, if you will, where there's a presence or an absence of a particular SNP. It signals the researchers and doctors for the presence or again the absence of a certain gene or a certain medical condition in the patient. So if you come across that SNP, you can be fairly certain that that person could be predisposed to a certain medical condition. If you don't come across that SNP, you can put that person into another category and say that you are not, luckily or unluckily, you're not likely to contract that kind of a condition. So in the case when gene happens to be, when you find these SNPs, when you find these markers around the gene that happens to be a faulty or a mutated one, you can utilize these SNPs for disease diagnostics, right? So again, you stumble across that gene, and you stumble across that SNP, and you also stumble across that SNP in the population that is heavily affected by by a certain condition. And if you take your patient, you find that SNP in him or her, you can state, okay, you have a certain probability to develop a certain condition, for example, high blood pressure or maybe cancer, just because we found the SNP that is closely associated with this medical condition in other people. So the conditions where the SNP is directly involved and responsible for the development of the disease, I usually refer to as monogenic or single gene conditions, for obvious reasons, because you have a single gene involved in the development of the phenotype, whatever that phenotype happens to be, drug sensitivity or cancer, sorry, not the cancer, I guess, bad example, sickle cell anemia. So in this case, you find that SNP affects the structure of the protein, that faulty protein fails at its functionality and leads to a specific phenotype, faulty phenotype. So a very easy, somewhat easy case where one SNP can be attributed to one gene and one gene can be attributed to be responsible for the whole condition. You fix the gene somehow, or you at least you identify that gene, you are now have a strong link between the SNP inside of that gene and the medical condition. While for so-called polygenic conditions here, where conditions where multiple genes, as well as not just the genes, but other factors involved in generation of the final phenotype. For example, all sorts of environmental pressures, physical activity, for example, dietary restrictions, and so on and so forth. For these kind of conditions, direct identification of the SNPs involved with the condition is quite often unfeasible or just straight up impossible, because you have, first of all, you have multiple genes involved in the development of the, or responsible in the development of the condition. And after all, you cannot be absolutely certain that you have identified all of these genes, just like it is the case with cancer. You can't really pin it down in a lot of cases to a specific gene. And not to mention what makes them even more complicated, like I said, all sorts of environmental factors affecting the onset and the development of the condition. So finding one SNP that is responsible for everything in those kind of polygenic conditions is quite often just straight up impossible, like I said. And unfortunately for us, such polygenic conditions are actually making up for the majority of the most troublesome diseases that we know, like cancer, diabetes, variety of heart conditions, deep veins, thrombosis, asthma, and some other ones. So even though identifying, specific mutations, specific SNPs as immediate, direct, causative agents, immediate, direct reasons behind these conditions is extremely hard, if possible at all. Quite often, like I said, it's impossible. We can still, nevertheless, at least exploit these SNPs for predict the predisposition as well as the susceptibility of the patient to such conditions. So we can't really fix it, but at least we can say that if we find these SNPs, we can tell that person in advance that you are running a risk of contracting that condition or developing that condition at some point in your lifetime. And we're only predicting, we're not really certain. We cannot claim that we established that link with 100% probability. We'll talk about this a little bit more. So we're only predicting, we're only kind of assuming it might happen with a certain degree of certainty. And that SNP, that marker SNP, may or may not, and quite often, almost always, it actually does not have any functional impact on the, on the onset of and the development of this condition. So it is straight up just a marker. And the simple rule of thumb here is how do we, the long story short, how do we use these SNPs as medical markers? The very simple rule of thumb here is the further away from the gene the SNP is located, the less accurate your association between the gene or the genetic locus in general and the SNP will be. So again, the further away that SNP is from the gene or from genetic locus, the less accurate your association between the presence of that SNP and the patient's predisposition to a certain condition will be. The closer the SNP is to a putative gene causative agent for a condition, the more accurate your association between the gene and that condition is going to be. In other words, the shorter the distance, the higher is the correlation between a medical condition, say cancer, specific type of cancer or whatever, and patient SNP profile. The shorter that distance, the higher is the correlation. The longer the distance between the SNP and the gene, the less accurate your correlation. The weaker is the correlation. Notice, by the way, that I'm using the SNPs and the SNP profile terms kind of interchangeably. I will explain this shortly. It's not just it does have the reason for it. It's not the same thing. The SNP and the SNP profile. I will explain this in a bit. Just a long story short, I guess it's usually it's not one particular SNP that scientists are trying to link with a specific condition. It's not just one nucleotide, right? It's not just one variant. Like in this case, you have two people, and then this guy has T in the position, and that, let's say, is indicative of some sort of a disease. So quite often, it's not just one single nucleotide that is being used as a marker, but it actually a set of SNPs, an array of SNPs, if you will, a certain pattern of them, multiple SNPs arranged in a certain patterns, as they call them. And those patterns are usually referred to as haplotypes, and we will definitely spend more time studying that. So using those haplotypes, using those sets, patterns of SNPs, as opposed to just one SNP, quite often allows you to predict the association between the SNPs and the medical conditions a lot more accurate. You can think of a simple analogy here that I think should kind of bring home the message. For example, if someone was asked, to describe the random person that he or she randomly met to you, right, you'd probably find the description based on multiple traits of that person. For example, height, the eye and the hair color, what kind of clothes the person was wearing, whether the person was wearing glasses or not, and so on and so forth. So that kind of description using multiple traits would be probably a lot more accurate than if someone just gave you the eye color of that individual, right? So it's kind of the same logic here takes place here with the SNPs and the links, the associations between the SNPs and a particular genotypes. Using one SNP, sometimes it's doable, sometimes it's possible, but quite often it's much easier for multiple reasons that we will go over soon enough. It's much easier to use multiple SNPs or patterns of SNPs and link those to, or try to link those patterns to genetic conditions. The next two slides are just simple illustrations of the simplest cases behind the SNPs affecting individuals' phenotype. So those kind of cases that I described as monogenic conditions. So when one or possibly several SNPs are directly, and that's a key word here, is directly responsible for the structural change within the protein. In this figure here, for example, you have some sort of a protein, whatever it happens to be, doesn't matter. And you have two SNPs highlighted on the side here in both variants of this genomic DNA. So this is the first one, and this is another sample. And the SNPs, the mutations are highlighted. So as you can see, G in the first variant gets substituted for A, and T also gets substituted for A in this case. And what happens is in this simple monogenic condition, again, when one or several SNPs directly impact the functionality of the protein via its structure. So what happens in this case, you get these mutations and potentially, not necessarily, but potentially an active site of the protein has at least two, right, amino acids substituted for some other amino acids, some other ones. which leads to, from what it looks like a significant structural change, right? You have a completely different, from the protein standpoint of view, from the functional standpoint of view. This kind of a change, this kind of change of the structure is kind of important. It's not negligible. So it leads to these two substitutions, these two mutations, these two SNPs lead to a significant change in the structure of the protein. And like I said, imagine that this was your active site of the protein. So clearly, any structural change here may lead to, drastic alterations to the protein's activity and functionality, and that can be manifested on the phenotypic level, um, which can be tracked. So for the next slide here, it's very similar to the previous one in its essence, but and it has to do with the lab that we performed a few weeks ago, right before, in the pre-COVID-19 era, you know what I mean? So you have your receptor here in your tongue, which, depending on the structure, and the structure is governed by the DNA sequence, and specifically the presence or the absence of the SNPs. So this protein is either capable, like in this case, or incapable of binding the PTC molecule, the molecule that we perceive, that we taste as bitter. So as you can see, some people have this weird kink here on the surface of the receptor, and this precludes the docking of the chemical onto the receptor, which obviously manifests itself in your inability. So there's no signal. This is a signaling molecule, so there's no signal going out, and you perceive it as inability to taste this chemical. You don't taste that it's bitter. A different SNP in this position again, and these are one of those examples of the monogenic conditions, the simplest ones. Doesn't mean necessarily that they are the simplest from the medical standpoint of view, but they are much easier to identify. So this particular one is not a disease. Again, don't get paranoid. It's just a funny phenotype kind of thing, but it is definitely belonging to this monogenic conditions. So a different SNP in the genomic DNA sequence, encoding, for that receptor results in, as you can see, the absence of the kink, and hence, successful docking of the PTC onto the surface of this receptor, which leads to the activation of the receptor, activation of the signaling cascade, and ultimately your ability to taste that chemical and perceive it as bitter. So again, very important to understand. These are the monogenic conditions, not necessarily medical. I don't know what kind of protein this is. It's just a hypothetical protein. This one here, your ability or inability to taste the PTC has nothing to do with your, biological fitness or anything like that. So, but the key point is that specific SNPs in these positions, governing the structure of these regions of the protein, the SNPs are directly involved in the functionality or the change of the functionality of your protein, of your receptor. Okay, so that's that. So moving on, some more facts about the SNPs, or rather, I guess most of these facts were reiterated multiple times already throughout the lecture. So SNPs found, so a variation to be considered a SNP, it must occur in at least 1% of the population. So we discussed that. Again, I'm not going to spend time on it. Make sure you know this value. SNPs can occur in both coding and non-coding regions. So the ones that occur in coding region, those are the ones that are usually attributed to monogenic conditions, and the ones that are occurring in the non-coding regions are the ones that are usually being investigated for their potential to be genetic markers, right? So, and that's what most of our discussion throughout the rest of the lecture will be concentrating upon. Because only about 3 to 5% of the person's DNA sequence codes for the production of the proteins, most SNPs are found outside of the coding sequences. Again, we already discussed that, not going to stop here. SNPs found in the coding sequence are of a particular interest to the researchers because they're more likely to alter the biological function of the protein. So again, particular interest here, this part here. It is of a particular interest, don't get confused here. It's a particular interest. Um, Definitely, these are one of the most interesting ones. Definitely, these are one of the most interesting ones, but they are of a particular interest, I would say, more so to the pharmaceutical companies who are looking into immediate profit from these findings, right? And only because it's what we call in science sometimes a low-hanging fruit, right? Once you identify such a SNP, you link it to a specific condition very easily, and um, given that most of the time you'd be looking at a monogenic condition, the SNP will comprehensively define the condition and can be exploited. In a good way, exploited here, obviously, for screening of the disease, maybe potentially in the future for fixing that disease, or something that is more likely to happen nowadays when you know that SNP, you know that that SNP is responsible for the functional change in the protein. You isolate the protein, you crystallize the protein, you look at the structure of the active site affected by that SNP, and what you do, your next step is you try to create a drug. You try to come up with a drug that will specifically target that specific altered active site of the protein. So that's definitely something that is being, you know, being done these days. Um, Most of the modern day research, though, clusters around actually the SNPs that are not part of the coding region. So they don't fall into the coding region, since those medical markers are absolutely invaluable for in our effort to, you know, decipher and um, treat eventually such complex conditions, medical conditions like diabetes, cancer, whatever, heart conditions, and the rest of them. So definitely a lot of interest is dedicated to these SNPs. Uh, this one here, SNPs are also being evolutionary stable, so they're not changing much from generation to generation. That's definitely an important statement, very important concept you need to understand. I don't think we discussed it yet. So SNPs being evolutionary stable, right? Why is that the case? Ask yourself, maybe pause the video, try to answer that question yourself. Why do you think they are evolutionary stable? How do you explain this phenomenon? So the reason for the SNPs to be evolutionary stable stems from the fact that, as you know by now, most of the SNPs lie outside of the coding regions, right? So outside of the actual genes. And for that reason, they are not necessarily manifesting themselves, right? Sometimes they are in a kind of tricky, weird ways, but most of the time they are not manifesting themselves. And therefore, if they don't manifest themselves, if there are no phenotypes associated with those SNPs, they are not necessarily the primary subjects to natural selection, because there's nothing to select for, right? Or almost nothing to select for if the SNP doesn't manifest itself phenotypically. So that's the reason for it. And if you think about it, however, it is also a very handy property of the SNPs from the research standpoint of view. Because if you use SNPs, if you plan on using those SNPs for the complex conditions as the road signs, so to say, as the medical markers, right? Uh, when you rely on predicting patients, you know, predisposition or susceptibility or just straight up response to a certain disease or the drug, you absolutely don't want your SNPs to be unstable genetically, because if they're unstable, you wouldn't be able to rely on them. Just like with the road signs, right? Think of the direct analogy. What would happen if you had your, you know, road signs change on a daily basis, on a weekly basis, or something, right? So that's not a very handy road sign. So exactly the same logic applies to SNPs. You want them to be genetically stable, so that if you, you know, spend all this time and spend all this money uh, to identify, to associate certain SNPs to certain medical conditions, you can be, you know, can be fairly confident that you can apply this methodology not just to one person, but to, you know, hundreds and thousands of people out there, so that every time somebody comes to the medical office and asks to be tested, for example, for their susceptibility to a specific form of cancer, people have these stable SNPs that they can screen the person for. The person has the SNPs, you claim that there's a certain probability. Again, never 100%, but there's a certain probability of that person developing that cancer. If the person is lacking those SNPs, you say that you're unlikely to get that condition. Again, doesn't necessarily mean that you will never get it just because you don't have the SNPs, but the chances are definitely lower. So clearly for that application, I hope you got it from this explanation. You absolutely want the SNPs, just like the road signs, you want them to be evolutionary stable.